Colony PCR Part A, and Part A is actually running the polymerase chain reaction, and then after lunch, then we will do the gel electrophoresis. So our learning goals for doing this particular lab is to learn about polymerase chain reaction. This has been a revolutionary tool in molecular biology, and so it's really important for students to have heard of it and understand the applications of this. So just really briefly, we're going to do this now, do the PCR lecture implementation. Then we're going to have a garden tour today, as well as a lunch. So, and then we'll run the gel afterwards, and then have our guest speaker. So I wasn't sure how much do you all teach this as part of AP Biology now, PCR?
Yes.
Yes, okay. So anyway, in case you haven't done it, this happens to be a revolutionary technique, I think. It's to target and copy a very specific DNA in a very large mixture of DNA molecules. And it's basically doing DNA replication in vitro, in a test tube, okay? Carrie Mullis invented this in 1985 and then got the Nobel Prize for this in 1993. And the reason I keep saying it's revolutionary is because I was in graduate school before this was invented, and back then, we didn't have these machines, these automated machines, and in order to clone, it took a graduate student like 6 years to clone a gene, sequence it, and then publish it, and that was your PhD. That was what we did. But so what we did in 4 years, what I did in 4 years, can now be done in 1 day. Okay, if you knew exactly what target you're trying to go for. So it has really changed the pace.
It's the equivalent of like when your grandparents said they walked 5 miles up the road.
Yeah, okay, all right. But it was true.
She's old enough.
Thank you. Okay, so it means I'm old. That's the other thing. Okay, anyway, so I'm trying to give you some resources. I do think the PCR animations are really, really good. I was thinking I might even show you one, but anyway, so the big concept is we need to have an idea of what target you want to go after. So that's a targeted gene. And in order to get that one part of the gene replicated, you must have a starting point. Okay, remember in DNA replication, you always have to have a primer before the DNA polymerase walks on. So typically in real cell biology, your primer is made of RNA, but in this case, we're going to use made cDNA. We're going to make DNA primers, and they're going to be going on the front end of the gene that you want and on the back end of the gene that you want. And so they're going to bind, and so that's the primers.
Quick question.
Yeah.
So you're saying that naturally primers are all RNA, but
Yes.
what we are engineering is that the primers will be DNA.
Yeah, the primers
Okay,
that we give you are going to be
Thank
DNA.
you.
Okay, and the main reason is because DNA is more stable than RNA.
Got it.
Okay, so anyway, so typically you teach, DNA replication, and you have the template DNA. Obviously, both strands of DNA are going to be replicated eventually, but we start off with the template DNA. Template DNA right here, 3 to 5, and then you're going to bring in. Then you're going to bring in the DNA polymerase. So that's the enzyme that makes the DNA polymers. They're the enzyme that will join 2 nucleotides next to each other, make that phosphodiester bond, and they only work a certain way, right? DNA polymerase only walks from 3 to 5. All right. Okay, so we know that that's what happens in our cells, and we are going to try to replicate this. So the key discovery was they went to the, Yellowstone National Park, where the Old Faithful, the geyser comes out, right? And there's hot water, and there happens to be bacteria that live there, and they were able to extract those bacteria and pull out the DNA polymerase that bacteria cells use. Okay, that bacteria is called Thermus aquaticus. So now we call this DNA polymerase capital T, little aq. So Taq polymerase stands for the DNA polymerase that comes from that bacteria. There's now a sign on Yellowstone National Park biology teachers are not allowed to scoop water out of Ofeifo.
Were they doing that before?
Oh yeah, yeah, of course. As soon as we find out this can be done, oh yeah, we go there. You can actually, I met a teacher who actually goes to his hot water heater at home and he pulls bacteria from his hot water and has it purified for the students. So anyway.
And that was in his hot water at home?
Yeah, because this particular type of bacteria is adapted to living in very, very hot conditions. Yeah, so you can find it probably in your home if you go look. Anyway, so by having this one DNA polymerase, by having this one DNA polymerase that's able to withstand hot temperature, it revolutionized the idea of how this is going to work. And now we also have computer. I can't think of what the word is, but the computer cyclers that actually can program the temperature really quickly. Okay, so the emission of the thermal cycler with the microcomputers for programming the temperature. So we're going to have 3 different styles of the thermal cyclers to show you, and the machines are going to be automated, so it's much quicker. So the big machine that I used to use when it first came out, my first big PCR machine was like this big. Okay. So back then, so I think 30 years ago, it cost me $30,000 to buy that for my college class, and I really had to do a lot of convincing that it was going to be worth it.
You're out the deal.
And, yeah, and then now, this is my favorite one. It's called the Mini One PCR, and it can fit in my backpack. It can fit with my gel electrophoresis case, in the carrying case, and it can go off to do whatever. Okay, so. Yeah, when I first started in graduate school, we had three hot plates, 3 coffee pots to do 3 temperatures.
Oh.
I'm not fully familiar with PCR. Why is heat involved?
Okay, I'm going to show you right now.
Oh,
okay.
okay.
Yep.
Oh no, I was going to mention about those hot plates. I
Yes.
remember doing an internship with my professor, my mentor. He still had the mechanical arm with 3 hot plates.
Yeah,
So that mechanical arm would go
Okay,
back and
and
forth.
he was even better because I had to stand there with a timer. 1 minute, go.
She was the mechanical arm.
Yeah. And you do that 30 times. Okay, so if you teach DNA replication in detail in your classes, it involves 10 different proteins. Okay, so you have to have the helicase to break open the helicase and then separate the 2 strands, right? Then you need to have the primers come in. Then you need to have the RNA polymerase, and you need to have the DNA polymerase 3. Then you have to have DNA polymerase 1. Then you need to have the ligase. Okay, so there's all of those things. Okay, all right. So we don't want to put all of those into this test tube. So the trick is, if you heat your DNA to 95 degrees Celsius, then that will break the hydrogen bonds between the complementary base pairs, and it'll go from 2-stranded to single-stranded. So that's the first step.
Okay.
Oops, I'm sorry.
Oh, so that eliminates the need for those extra enzymes. It's just
Yes, and
already-
you don't need to have single-strand binding proteins.
Oh.
The heat is going to keep them apart. So that's the very first step, 95 degrees Celsius. Obviously, we can't do this in our bodies, right? We are only at 37 degrees. So we can do this in a test tube with high heat, and it doesn't destroy the DNA. It just breaks the hydrogen bonds. Okay, it doesn't break the covalent bonds because you don't want that. It only breaks the hydrogen bonds. Okay, then the next thing is, well, we know we need to have primers come on to the DNA, and the complementary base pairs have to form hydrogen bonds. So obviously we can't have it at 95 degrees anymore. We need to cool it down so the hydrogen bonds can form. So that's the second step. That's called annealing, and that's when the primers find their find the correct sequence on the template DNA, hybridize, form those hydrogen bonds. Okay, annealing means that. Okay, then the third thing is, now once you have the primers on, you need the DNA polymerase to come on and start not putting down, the nucleotides are all floating around. The single DNA nucleotides are floating around, but when they find the correct one, the DNA polymerase will join them together and walk along, and that's called the elongation step. Okay? So because we got this DNA polymerase from thermo aquaticus, it can work at a higher temperature. Higher temperature will be more efficient, and so 72 degrees is why you do the elongation at that temperature. So having those 3 temperatures makes this one replication process occur, okay, without all those other cell proteins involved. You just need to have your Taq polymerase. You give them DNA primers that you want, targeted against the gene that you want. You put in all this single DNA nucleotides, plenty of them. You give them the cofactors from that for the DNA polymerase, and then let it go, okay? So if you go through one cycle, 3 steps here, high temperature denature the DNA strand to single strand, anneal, the primers come on, and then to do the DNA elongation or synthesis, you've now basically copied that one little stretch of DNA. So what happens if you do it again?
And you got quadruple.
Then you'll get quadruple.
So the annealing of the primers, you mentioned before the primers are DNA
Yes, in this
Okay.
case, not like
in
yourself.
this case. So then, can you repeat again why the difference between 57 degrees for the second step versus 72 for the
Okay.
third step if they're both DNA?
Oh, it's just, the 57 degrees is just to make sure that the hydrogen bonds form very well.
And the
Between,
72s for the...
for the DNA polymerase to work a little bit faster. You
Oh,
just
it
want the
just speeds it
process,
up.
it just speeds it up.
Okay.
You want enough time for the primers to find the target and make those hydrogen bonds. And you need them to stay there, right?
Yeah.
If you put it too high, maybe the hydrogen bonds are just really super loose because it
Okay.
keeps popping off. So you want to cool it down to make sure they're there, and then have the enzyme come in and then go from there.
Okay, thank you.
Yep. Okay. All right. So then, okay, this is a animation from DNA Learning Center. Can I show the videos? Okay. I think it's only 3 minutes long, so I'm going to show it to you because they do a good job with all the animation.
Oh, I didn't know there was a video.
And cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence. The enzyme Taq polymerase, shown here in blue, binds to the prime sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
So you just dump a bunch of Taq polymerase in there and...
Yes, we give them extra of everything.
Okay.
Yeah. Okay, great. So this is a really quick overview for the students. And then I put in slides in case the video didn't work. So anyway, so what we mentioned before, I did a little bit more detail for you. So this is the denaturation steps to break the double bond to single bond, single strand DNA. And then the primers anneal. So we have both the forward and the reverse primer, it's called, to surround the gene of interest that you want. So you can get replication on both strands. Okay. Then here's the elongation of the DNA. And again, because the DNA polymerase only walks factor 3, or it's going to elongate factor 3, you're going to end up with 2 DNA strands. And then, so the first cycle, you make 2 molecules of DNA, then you make 4, then so on and so forth, okay? So after, I put put a graph in. So after 20 cycles, you can get 1 million copies of DNA. After 30 cycles, you get 1 billion copies, okay? And so you could do the math if you want. If you had, 1 minute at each of the temperatures, how long would that take you? And it really is a matter of how fast can the computer talk to your heater or the cooling system in that thermocycler machine to bring it up to the different temperatures, okay? And you'll be able to see that with the big Biorad machines, it takes a little bit longer than the Mini One, because the Mini One has this Peltier heating cooling system that I don't understand, but it goes much faster. Anyway, yeah, with that machine I can do it like in an hour and a half. Okay, so why are we even doing? Did you have a question? Go ahead.
I did. I want to ask about the overhangs.
The overhangs. Oh, yes.
So one is, why do they exist in the first place? And then the second question is, why after like a few, like after the first couple of cycles, then it stopped replicating the overhang?
So the overhang is because you got the genomic DNA, right? You have a long, long piece of genomic DNA, and you're only going to go after this one little target gene.
Mm-hmm.
So like if we were trying to do PCR of human cells, we would have 3 billion nucleotides, and I only want to target 2,000 of these of the nucleotides in that one.
So you're just saying, like, in the beginning when we first cut it, it was just longer than what we needed.
Yeah.
That's why it was there. Okay.
Yeah, or however, sometimes we don't cut the DNA beforehand, we just throw the whole thing in and then just, but the primers will find the target that we want.
Oh.
Yeah.
So that's why the first time, because the primer
hang.
is like a certain orientation, so it will still do the other overhang, because it would go all the way there. But then the next time it finds it, it's here, so then it'll be the right length.
Yeah, so that's when you have a forward primer. It's going to go one way on one strand, and the reverse primer starts the back of your gene and goes the other way.
And so it will still have a little bit of overhang.
Oh, it will definitely have the overhang on that, the original.
But then the next cycle.
But the next cycle,
is
you only have this. Yeah. Oh, right. Very good. Yeah. So by the time you get 1 million or billion copies, that one extra copy doesn't matter.
Right, and then it wouldn't show up on the gel
anyway. Yeah.
Okay.
Yeah. Very good question. Okay, excellent. Anybody else have questions about the idea of PCR? Okay, the concept of PCR. So again, we're trying to have your students learn about PCR because it is such a valuable tool now for molecular biology. And I think I put in a slide later for applications. And so we're going to test it in your plates, your agar plates, because some of you got to see that you have both white colonies and pink colonies. And we want the students to really understand genotype goes to phenotype, right? Why does one bacterial colony have white and why does one colony appear pink? So you all know the answer. What's the difference?
Different. They don't have the gene for the RFP.
Okay, so it could be the bacterial colony does not have the gene for the RFP. And is there any other reasons?
Did it take up arabidose?
It's backwards.
The gene was backwards, okay. Yeah, or there was no plasmid in the bacteria, but you're not really testing your untransformed cells, so yeah. So we're going to be looking at the transformed cells, right? So we're going after the ABE-LA plate, the 3-stripe plate, okay, and that's where you saw both pink and white colonies. And again, we're very lucky because we are able to see that red fluorescent protein express. Again, in humans, typically, most of your proteins are not visible. They're clear. And so, we have to have a way to figure this out. But I do love the idea of teaching genotype to phenotype to students. Okay, so red colonies or the pink colonies should contain that P-era-R plasmid with both AMP-R and the RFP genes, as you mentioned. And then the white colonies should only contain the P-era plasmid with AMP-R and no RFP genes. Okay, and so that's what we're going to try to do. And anyway, a little while ago I mentioned all the things that the PCR actually needs, and Karen calls this the secret sauce, right? That we have to give our DNA tubes the secret sauce, and it will contain this, what we call the master mix, okay? And that master mix must include that DNA Taq polymerase that came from Thermococcus. It must have magnesium chloride because that is the cofactor for most DNA polymerases. It must have a whole bunch of deoxynucleotide triphosphates, so those are the single nucleotides, free floating. Every enzyme always has to have the buffer to keep the right pH and the salt concentration. And then we're going to put in some glycerol to make sure that the sample sinks down and works together. Okay, so that's your secret sauce. And this is actually a very critical step for you as a teacher to prepare this. And so Karen will give you more tips about this. But basically, you're going to be getting primers, and you have to thaw them. Then you will be also be getting master mix, and you're going to add your master mix with the primers together before you pass it to the students. Okay. And because when you run the PCR, I mean, we'll go into more implementation, but anyway, you try to make the correct amount of master mix and primers, because once you mix it all up, you can't refreeze it and then reuse it at a different time. Okay, it's whatever is done is done. So you only do it for that one class. Don't make, I think that's true. Well, Karen can tell you everything. And then you keep it on ice, and then only mix, what you need for that class. Okay, Karen, did you want to show them, or can I just do it on the, you want to show them?
Sure. So on your table, you have a cup with a tube labeled PCR, and that's what the guide is calling our master mix. It's the master mix itself plus the forward and the reverse primer. And you have 4 PCR tubes, and then you've got your colony. So what you're going to do, you have, the aliquoting guide tells you to aliquot 96 microliters into that tube, the PCR tube for the students. And then you're going to put 23 microliters, they are going to put 23 microliters into each of those 4 tubes, because they're going to run 4 samples, right? And what you're actually going to do is, the first sample is going to be some of your red cells, the second sample is going to be some of your white cells, the third sample is going to be a control plasmid, a firm red, and the fourth is going to be a control plasmid for the white, the PR. And so when you want to inoculate these, let's put some of your, maybe P20s.
Oh, the top
Put away.
P20s.
Oh, okay. Oh, I didn't see, okay. So let's put some of his into one of his tubes. So we're gonna put, I guess they need the P200s, because they have to be 23 in each tube.
Okay, nice.
So just to make this enough.
So turn this off or keep it on?
Yeah, sure.
Yeah, but